Now that you've watched that lovely crash course on macromolecules, I can't resist critiquing Hank. In his jar of peanut butter, the main macromolecule in the solid part is protein from the peanut paste. He made it sound like a hunk of lard, all fat. It's not. Next up, lipids do not make cell walls. They do make up cell membranes. We'll go into more detail on the difference in later lectures. And lastly, I can hear nutritionists all around the globe screaming in pain at his statement that all you need to be healthy are the 3 macromolecules. We'll be talking about vitamins and minerals later when we talk about enzymes. I'll leave the discussion of fiber, phytonutrients, and such for any nutrition classes you may take. Next up is my take on nucleic acids and what is important about them. We will be dealing with DNA and other nucleic acids a lot in the course, so make sure you have a good basic understanding of them. On to the nucleic acids. As far as structure, nucleic acids are made up of nucleotides. These are the monomers of nucleic acids. Nucleotides are composed of a 5-carbon sugar, a ribose, and you have a nitrogenous base. See the nitrogens. We generally abbreviate that as just a base. And you can change these bases to make the different nucleotides. There's 5 different bases. We'll talk about those later. And then you have a phosphate group. Together, the phosphate group, the sugar, and the base make the nucleotide. Now let's talk about the functions of the nucleic acids and nucleotides within the cell. These 3 groups, ATP, NAD+, and its other form, NADH, FAD, FADH2, are involved in energy transfer within the cell. You've probably heard of ATP, and when you lose, well, let's look at ATP. Here is ATP. We have our ribose, we have our adenine, and together, this is adenosine. Now, when you put 3 phosphates on, it's adenosine triphosphate, or ATP. You can clip off this phosphate group and move it to a molecule, and energy goes with it. And this goes from being ATP to ADP, adenosine diphosphate. So energy moves with the phosphate. We can take that phosphate, put it back on to take ADP back up to ATP, and it's like putting money in the bank. Now with NADH plus and NAD plus and NADH, FAD, FADH2, these molecules function in transferring energy around the cell by moving hydrogens around. Now these hydrogens, just like with the phosphate group, when we move it from ATP to another molecule, if you move a hydrogen from one molecule to another, energy goes with it. Now, DNA and RNA function in information transfer within the cell. They transfer information on how to make proteins from the genome to the rest of the cell. And we'll be talking about that in more detail. Let's look at the structure of DNA. Now, you take your nucleotides and you hook them together by, you guessed it, dehydration reactions between this hydroxyl group and this phosphate group, and you make a string of nucleotides that we call a nucleic acid. And in this figure, it's represented by this purple ribbon. We call this the sugar phosphate backbone. Now in DNA, you get 2 of these strings together, and the bases hydrogen bond to hold this together in the famous double helix of DNA. This textbook doesn't go over the properties of water. The properties of water apparently are not necessary to understanding the biochemistry of the cell, but I needed to understand it before I understood phospholipid bilayers and the like. So what follows is a discussion of the properties of water. We will be talking about hydrogen bonds, hydrophobic interactions, solutions, acid, bases, salts, pH, and buffers. Let's talk about water. Water, as you may know, is 2 hydrogens bonded to an oxygen, H2O. Oxygen has an element number of 8. That means it has 6 electrons in its outside, or valence electron shell. It wants 2 more electrons. Hydrogen wants one more. So the two hydrogens get together with the oxygen and shares with the oxygen. Now, because oxygen is bigger than carbon, it can keep the electrons a little bit longer instead of stealing them. And so what happens is the electrons zip around hydrogen once, twice around oxygen, once around hydrogen, twice around oxygen, once around hydrogen, twice around oxygen, so that what happens is the molecule gets a slight positive and negative charge. Oxygen gets a slight negative charge because the electrons are spending more time around the nucleus of the oxygen, and the hydrogens get a slight positive charge. We call this a polar covalent bond. Think of it as the poles of the Earth. The Earth is a big magnet, and it also has a negative pole and a positive pole. Now, because water has a positive and a negative side, if you will, positive and negative pole, it forms bonds with other water molecules. The negative oxygen is attracted to the positive hydrogen, and so on and so on. And we call these hydrogen bonds. Don't get confused between the bond, the covalent bond, the nonpolar covalent bond between two hydrogens in gaseous hydrogen, and the hydrogen bond between molecules. Now, hydrogen bonds can form between any molecules where there is a hydrogen bonded to an oxygen or a hydrogen bonded to a nitrogen. Those are the ones that you need to worry about for this class. Now let's talk about what happens when you mix water with molecules that are not capable of hydrogen bonding. Here we have an example of a string of carbons, non-polar covalent bonds between the carbon and the hydrogens that are attached to it. And we have a bunch of waters. Well, the waters, because of their polar covalent bonds, they've got the slight positive and slight negative charges. They're attracted to each other. So this one comes over and says, hey, I can form a hydrogen bond with you, and it's all happy. And this one over here is saying, oh, well, don't leave me out of the party. And the carbon molecule is saying, hey, you're stepping on my toes. And it keeps getting more and more crowded. Hey. And it can't form hydrogen bonds with the water because it doesn't have an oxygen bonded to a hydrogen, nor does it have a nitrogen bonded to a hydrogen. So what happens is it's excluded by the water. Now we call this hydrophobic interactions. Molecules that are incapable of forming hydrogen bonds are excluded by the water that wants to hang out by itself. Okay? Kind of sounds like cliques in high school, doesn't it? Now, molecules that can form interactions, hydrogen bonds with water, are called hydrophilic. Philic meaning loving, hydro for water, so they're water-loving molecules, whereas molecules that are excluded by water, that can't hydrogen bond, are called hydrophobic or water-fearing. If you take salt and put it into water, what happens? It disappears. It dissolves. If you taste the water, it's still salty. The salt is still there. What's happening on a molecular basis is the water, because it has a slight positive and a slight negative end, can interact with the ions of sodium chloride. Okay, so here's what's happening. The sodium ions say, hey, hi, water. And the chloride ion says, hey, hi, water. And they separate. We call this dissolving. Okay. Now, water can do this because of its slight positive charge and slight negative charge. It can also do this with molecules that are hydrophilic. Okay. So, for example, sugar is not an ionic compound, but it does have oxygens bonded to hydrogens, so it can do the same thing. So when you add sugar to water, it dissolves. Okay. Now, we call this a solution, either a salt solution or a sugar solution. The solid stuff that you put into water we call a solute, and we call water the solvent. You put a solute and a solvent together and you get a solution. Now, water is important because it's the universal solvent. It can dissolve most things. And your cells exist as a water solution. So it becomes very important in biology. From here, we're going to discuss what else happens in different types of solutions. There are two types of ionic substances. Those like hydrochloric acid that release hydrogen protons, hydrogen ions, into water when they dissolve. We call this an acid. That's why we call it hydrochloric acid. There's also those that when they're put into water, they release a hydrogen-oxygen bonded ion. We call this a hydroxide ion. And these have a tendency to combine with any hydrogens that are floating around in the water and form more water. We call these bases. So an acid is anything that releases hydrogens in solution. A base is anything that removes hydrogens from solution, namely if it has a hydroxide ion. Or, we'll get to this later on in this chapter, an ammonium ion. Okay. Now, if you combine an acid with a base in solution, they will combine and make a salt and water. These hydrogen and hydroxide ions get together to make H2O. The sodium and the chloride get together to make sodium chloride. We call that a salt. Both acids and bases are important in biology, as well as salts, since your cells are composed of both. And your cells are very busy keeping the balance between acids and bases. To measure whether something is more acidic or more basic, we have a pH scale. It's based on how many hydrogen ions, how many free protons are floating around in the solution. Now, this is a scale that goes both up and down. So let's start at the middle, or the starting point. This is where we're considered to be neutral, where you have equal numbers of hydrogen ions and, in this case, hydroxide ions, or ions that are taking hydrogen ions out of solution. This is a pH of 7. Pure water is considered to be a pH of 7. Now, as we go to smaller numbers or down the scale, unfortunately the scale has it going up, and as the pH lowers, you get increasingly more and more hydrogens. So if you look at the substances that are listed, as they get more and more acidic, you can recognize that. They get, when you taste them, more sour, more corrosive. Okay. So milk is just a little bit acidic. Urine is just a little bit more acidic. Tomato juice, it's kind of tart, unless you've got some nice garden fresh tomatoes. And wine, grapefruit juice, lemon juice, stomach acid is a pH of between 2 and 1. Now going the other direction, this is where you have fewer and fewer fewer and fewer hydrogen ions versus hydroxide ions or anything removing hydrogens. So human blood is just a little bit basic, or alkaline is another term for it. Seawater is a little bit more alkaline. Milk of magnesia, that's back, and most of you are probably not familiar with that. They still sell it, but use it for an upset stomach. So if you have too much acid, you drink something that's a little bit basic to neutralize the acid and create salts. Ammonia, you wouldn't want to drink that. Household bleach, wouldn't want to drink that. And you get oven cleaner, that's our sodium hydroxide that we had in the last slide, and lime water. Okay, now, your body needs to stay at a pH of just a little bit alkaline. So it uses buffers to maintain that pH. Buffers are salts, or more usually weak acids or weak bases that can adjust to an increase or decrease in hydrogen ions by either taking up hydrogen ions or releasing them. I was going to tack on my section about hydrocarbons, functional groups, more about carbohydrates, proteins, but I figured that was enough. I am going to include that information in a video that's titled Hydrocarbons and such, and you can watch it if you feel you need it. But in the meantime, we'll say we're done. And you may be feeling overwhelmed. It's also the most hated microbiology. Unfortunately, it won't go away. The chemistry portion of this course will be reappearing again and again and again. So make sure you have it. I love answering chemistry questions. Feel free to email me, get with me during my office hours. I love answering chemistry questions. There are also some wonderful tutors if you want somebody to explain it a different way. Sometimes that's the way that clicks. And repetition is a wonderful thing. The more you're exposed to the material, the more likely you are to have that aha moment of, oh, I get it. Why didn't I get it before? It's all about repetition and becoming familiar with the material. So, now you should be able to, or after you've watched through these several times and read the textbook again, you should be able to name the 4 main families of biochemical, provide examples of cell components made from each of the families of biochemical, explain primary, secondary, tertiary, and quaternary structures as seen in proteins, list the 3 main components of nucleic acids, name the nucleotides of DNA and RNA, and list the 3 components of ATP.